Thank you for joining us for this webinar in which we will share our experiences with the MACSima Platform. This in our journey to make spatial technologies more available for researchers within and beyond our institute. My name is Evelien, and together with my colleague Elina, we will host this webinar. First of all, I would like to give you an overview of our work environment. Our team is part of the VIB. The VIB is the Flemish Institute for Biotechnology that unifies 5 universities spread over Flanders. So it's really a decentralized institute. There are almost 2,000 people working in this institute, and we cover over almost 80 nationalities. Research in our institute is focused on 10 areas, going from plant system biologies to cancer research, microbiology, but also computational biology and AI. VIB Technologies is a department that supports the centers by offering services in technology. We do this via expert consultancy, collaboration, and training. So the focus is on technology, and we also engage in partnerships with instrument providers and industry. This for various technologies, going from bioimaging over sequencing to single-cell technology. Our team, the VIB Spatial Catalyst, operates under the umbrella of VIB Technologies. Our mission is to make spatial technology more available for researchers by offering consultancy, trainings, wet lab services, and data analysis support. Not only for spatial proteomics, but also for spatial transcriptomics and metabolomics and combinations thereof. We have a variety of instruments at our disposal, of which one of them is the MACSima Platform.
As a Spatial Catalyst group, we have been using MACSima in service for a couple of years already, and we would like to tell you more about how we do it in core facility environment. As Spatial Catalyst, we provide full journey support starting from the experimental design, including sample preparation, data acquisition, data processing, and data analysis. And to do all this, we need a multidisciplinary team, which we luckily have. Our team consists of our team lead Evelien, who is responsible for project intakes, experiment planning and design, QC oversight, and post-run evaluation. We also have 2 lab technicians, including myself, and we are responsible for antibody testing, sample prep, and instrument management. And finally, we have a team of data analytics, which consists of 2 software developers and 1 bioinformatician, and they are responsible for everything that is data related, starting from data transfer and pre-processing and finishing with user support and training and data analysis. What is also important to note is that before we get into the experiment design part, we encourage our users to contact us to discuss what is the best option for their spatial experiment. To get some more information on the services we provide, both internal and external users can reach us via the platform called Connect. Via this platform, users can also see how occupied each spatial platform is, including MACSima. However, the actual bookings can only be made either by us or by selected key users. Now I'd like to talk more about the actual experimental design for MACSima. MACSima is compatible with both human and mouse samples, which can be either FFPE or fresh frozen. As a Spatial Catalyst, we tested all of the types and they worked nicely for us. For fresh frozen, the appropriate fixation method will be either with PFA or acetone. However, it's very important to note that acetone fixation is only appropriate for shorter runs of less than 30 cycles because this fixation type doesn't preserve the morphology of the sample well. It's also important that if there is a possibility of samples being prepared for MACSima application, it is often the availability of antibodies that will determine the method of isolation and fixation of the tissue. When the sample type and the fixation method have been selected, it's now time to select an appropriate sample carrier for the tissue. Miltenyi has a range of Maxwell chambers to be selected from, and the choice will solely depend on the tissue size and the experimental design. Whenever it's possible, we always prefer to perform the sectioning ourselves. However, as we often work with clinical samples, we understand that it may not always be possible. So in cases like this, we provide users with slides with drawn area of where the tissue has to be placed on. We also always highlight that the tissue has to be placed somewhere in the middle of the slide, as ROIs cannot be selected too close to the border of the slide, as it will create some autofocusing problems. The slides that can be used for MACSima are either standard microscopy positively charged slides, and they have to be positively charged as they provide better attachment for the tissue, or high-resolution slides. High-resolution slides are provided by Miltenyi, and they are much trickier to work with as they are a lot more thinner than the ordinary slides. However, they do provide a better imaging resolution and sensitivity. It is also important to note here that high-resolution slides do provide a smaller imageable area even with the same chambers. Another important factor to consider in your experimental planning is run duration. Run duration will be affected by a couple of factors, and one of them being ROIs that are selected. The larger the ROI, the longer the total run duration. The run duration will also depend on the number of selected antibodies. The more targets, the longer the run. However, there is a possibility to optimize run duration by selecting ROIs wisely and taking into account the beam size for the bleaching. Also, optimal panel design and wisely choosing dyes can also influence your run time. For example, in our experience, we ran human VIA screen plates, which consists of 61 antibodies, and our ROI size was around 12 square millimeters. And in our case, it resulted in a runtime of approximately 3 days. And our longest run for a TMA sample finished successfully in 3 weeks. Now we are coming to the panel design step, which is possibly the most important step in the MACSima experiment. It will cost a lot of time, but it will be worth it at the end if it is done properly. So there are different antibody panel formats available. Option 1 would be antibody conjugates tested for mixed technology. Option 2 would be ready-to-use panel plates, which are also customizable. And option 3 is a combination of antibody conjugates and ready-to-use panel plates. How to choose antibodies and what are the key considerations? The first one will definitely be to understand the expression of the antigen in the tissue really well. What is the level of expression? What is the expression pattern in the tissue? Then it's important to understand which clones you're going to select, how specific they are, how stable they are, what is the format of these antibody clones. Then it's important to consider the fixation method that was selected for the tissue and how compatible it is with the antibodies that have been selected. It is also important to think of autofluorescent issues for certain tissue types and also for FFPE tissues and how compatible they are with antigen expression and antibody clones that are selected. And the last is fluorochrome conjugates, how bleachable, detectable, and bright they are. How to find the Miltenyi antibodies which are compatible with mixed technology? Here are the first steps. First step would be to define species and targets, then determine the best fixation method for your tissue, and then check the antibody availability at Miltenyi Biotec. However, this process can also be reversed, and the antibody availability will actually determine the best fixation method for your samples if there is a possibility to prepare samples specifically for MACSima application. To find the mixed compatible antibodies from Miltenyi Biotec, there are a few resources that can be used. First one is Miltenyi Biotec website. Second one is mixed antibody spreadsheet, which has been updated and circulated by our application specialists. And also antibodies can be found via MACSiQ view control module. MACSima is actually an open system which allows the use of third-party antibodies. There are just few tips and tricks to use the mixed compatible third-party antibodies. So the first one is that the antibody should be direct, although it doesn't have to be, and you can include some primary secondary combinations in your MACSima experiment. The antibody has to be conjugated to raisable fluorochrome. It also has to be suited for the immunofluorescence staining of fixed samples. And it is also preferred that the antibody was tested by the community and by other users. And there is an Excel spreadsheet which has a list of the antibodies which were also tested by the community as well. Third-party antibodies have to be optimized, and this is the general approach that we use in Spatial Catalyst. This approach we use outside of the MACSima instrument. Due to the interest of time, we don't want to occupy the instrument for optimization purposes. However, the same steps can be performed within the MACSima experiment as well. So for both FFPE and fresh frozen, we just follow the protocols of sample prep provided by Miltenyi. Then we block the samples in running buffer for 15 minutes at room temperature, and then depending on whether it's a directly conjugated antibody approach or primary plus secondary antibody approach, we play with some parameters including the dilutions and incubation times. Then we perform the DAPI staining for 10 minutes at room temperature, and then we evaluate the staining with wide field fluorescent microscope to find the best incubation time and the best dilution per antibody. When it comes to building an actual antibody panel, there are some general rules and tips and tricks to be followed to create an effective and a wise antibody panel. I'll just talk about the most important rules, and one of them is that all antibodies in the panel must be erasable to make sure that the signal is removed before the next cycle. It is also important to remember that the same fluorophore can only be used once per mixed cycle, and this is because antibody fluorochrome conjugates in the same cycle are stained simultaneously, and we want to avoid spectral overlap. It is also important to balance brightness with the marker expression. So highly expressed structural markers can be placed in the FITC channel. However, as I will mention in the next slide, if we can avoid FITC channel, this is the best way to go. Lowly expressed markers can be placed in the APC channel, and the intermediate or important antigens can be placed in the PE channel. It is also important to account for the tissue and fixation effects, as for example FFP tissues have a high autofluorescence in the FITC channel, so it is wise to avoid FITC in early cycles. Also for bone or lung tissues, there is also high autofluorescence, so avoid FITC at all or use for strong markers. And it is also important to manage spectral overlap, as FITC may bleed into the PE channel, and it is also might be required to titrate down antibodies for strong markers. So there are a lot of things to think about when it comes to the antibody panel design. However, we have always been getting a really good advice from Miltenyi. As I mentioned earlier, due to the acquisition photobleaching, the FITC channel should be avoided when possible. Otherwise, only use FITC channel for strongly expressed structural markers that are not useful for the quantitative analysis or segmentation, or minimize the effect by reducing the exposure time. So in our experience, this is the workflow that needs to be followed to build an antibody panel. It all will start with defining the biological targets. It can be done either using in-house experience, generated data, or publicly available atlases. Then the antibodies are selected, preferably from validated antibodies from internal or commercial databases. Then the antibodies will have to be optimized, either individually using different dilutions and incubation times, or this optimization can also be performed using RIA screen plates. It is also useful to include a control tissue to assess signal quality and background for each antibody. Then comes a panel design, which takes into account the balance in expression levels and spectral overlap. And then the panel is being tested and refined on the target tissue. For the final panel run, we also find it useful to include a control tissue such as spleen, which is positive for many markers, just to make sure that the run technically worked, and some of our users are actually doing this. In our experience, the biggest antibody panel we ran was 219 antibodies, which was part of the RIA screen Max Human PFA, with 14 custom antibodies for human liver tumor sample. Now I'd like to talk more about the actual sample prep and the tips and tricks that we use as a spatial catalyst. So in general, we strictly follow Miltenyi sample preparation protocols. They are easy to follow, very clear and fast. There is just slight modification that we introduced for FFPE sample prep. During the pernifization, we use 2 times 10-minute xylene incubations instead of a single 20-minute step, as this approach showed more effective paraffin removal. And this alteration was actually proposed by Miltenyi themselves, and this is why we are doing this. One of the technical challenges that we sometimes face, and not only for MACSima Platform, for many other spatial platforms, is tissue detachment, as certain tissue types and particularly clinical samples are more prone to detachment during pre-processing. So for MACSima, we also observed tissue detachment sometimes for particular samples. And to improve tissue adherence, we compared Nectarian slides and gold slides. And we found that gold slides demonstrated a notable improvement in tissue attachment. For tissue attachment, we also tested an additional baking step at 60 degrees for 1 hour immediately after cutting, but this alteration didn't improve attachment in the sample that we tested. Another technical challenge that we sometimes face is autofluorescence of particular tissue types. Some tissues such as lung, liver, skin exhibit stronger autofluorescence comparing to the other tissue types, and this can majorly interfere with MACSima imaging. So to reduce this background signal, these tissue types are photo-bleached prior to imaging, And for this purpose, we use VisGen Photo Bleacher, which is actually a part of a completely different platform, which we kind of repurposed for this part of the workflow. And we see that this helps to improve signal clarity and overall image quality. In our experience, lung is currently optimized using VisGen Photo Bleacher, and it requires 4 hours of photo bleaching. Liver is currently being optimized. Human neuroblastoma requires 3 hours of photo bleaching, and mouse neuroblastoma is being photo bleached using MACSima. So it is also important to say that photo bleaching optimizations can be performed within MACSima as well.
When the sample is ready, it can be loaded on the device for data acquisition. During data acquisition, it's advisable to regularly check the instrument, follow up buffer usage, and empty the waste. In case of errors, there's no need to panic. Often you can still rescue the run. When the data is acquired, we will export it to our network storage, as you can see here. Then the raw data is transferred to a local workstation where we will perform data pre-processing, QC, reporting, and data analysis. Both the analyzed data as the raw data are transferred to S3 storage. For data transfer, we use Globus. From the S3 storage, the end user can access the data and visualize it in a browser, this using a tool called Vitesse. When all the data is analyzed, we have a feedback conversation with the end user to discuss the results of the experiment. Let's dive a bit deeper into data processing and analysis and the tools we use for this. Pre-processing and QC of the data is done in MACSiQView. So we ask all our users to visually inspect the images to sort out the bad ones. And MACSiQView is great for this because it allows interactive exploration of the data. What do we mean with filtering of the data? Well, we want our users to evaluate each channel to check whether the antibody specifically bound to the targets of interest. Also, they need to evaluate if the signal-to-noise ratio is good enough. They need to identify potential artifacts in the images. And it's also crucial to check the bleaching efficiency in every cycle, because if there's remaining signal, it will interfere with the signal of the following cycle. So this step is very labor consuming and time consuming, but it's essential if you want to get good data out of your experiment. We already mentioned earlier that acquisition photo bleaching can occur and can have an impact on your images. What we do is manually annotate the acquisition bleaching artifacts to remove these regions from the downstream data analysis. For data analysis, we use Harpy. Harpy is a tool that's developed by Benjamin Rombout from the Ivan Sais lab, together with Arne de Vouw from the Spatial Catalyst. So Harpy is a Python package built on the spatial data framework, which organizes spatial data neatly on disk and in memory. It uses Dask to allow great scalability. Harpy integrates nicely with the entire SCVerse ecosystem, ScanPy, SquidPy, and so on. So Harpy, it's freely available, and it allows nucleus and cell segmentation, feature calculation, unsupervised clustering, and annotation. Harpy supports a large variety of state-of-the-art segmentation models, such as CellPose and InstantSeg, and the user can choose which model he wants to use for his analysis. Harpy allows you to calculate cell features. a wide variety of cell features like staining-based expression features, morphological features, spatial context features, and deep features. Based on these features, you can do cell type annotation and downstream spatial analysis, such as neighborhood analysis, spatial statistics, etc. Another option is segmentation-free analysis, also supported by Harpy. So you can do a pixel clustering and superpixel analysis. Enough background information for now. Let's look at some examples from the field. By now we have analyzed a variety of samples, neuroblastoma tumor samples, but also other tumors, TMAs, moya-moya disease, arteriosclerotic plaques, and so on. Also for mouse, we have been studying several tissues, liver, lung, and many more. First project I want to share with you is about neuroblastoma. Samples from a patient with high-risk neuroblastoma was compared to samples of a patient with low-risk neuroblastoma. Low-risk neuroblastomas typically respond well to therapy and in some cases may even show spontaneous regression. In contrast, high-risk neuroblastomas present significant therapeutic challenges, with relapse occurring in nearly half of all cases, despite intensive multimodal treatment. And there's more and more evidence pointing at the tumor microenvironment composition to be a critical factor influencing disease outcome. So these are unpublished results, so I cannot go too much into detail. But the main goal of the study was to localize cell types that are identified via single-cell analysis spatially. And it's important to use a complementary technique in this case, because single-cell data can be biased and enrich for lymphoid cells. So these samples were not only analyzed with MACSima, but as I already mentioned, a single nuclei RNA-seq analysis was performed on them, and also spatial transcriptomics. So based on the single nuclei RNA-seq data, together with publicly available atlases, a list was made of genes of interest, or targets of interest. These targets were compared with the targets available in RIA screen plates, but there was too few overlap, so very quickly it was decided to design a custom panel. The purpose was to be able to discriminate the cell populations identified in the single nuc RNA-seq data. So these markers were included in the panel, together with some general markers for proliferation, cell death, and so on. So in the end, a custom panel of 73 antibodies was designed. 56 of these antibodies were mix-validated antibodies provided by Miltenyi Biotec. 5 antibodies were Miltenyi antibodies, but were not validated to be compatible with mix. And then 12 antibodies came from third parties. Some of them were directly fluorescently labeled, while others were going to be used in a primary-secondary combination. Eventually, 52 of the antibodies worked. Here we can have a sneak peek at the results. So on the left, you see the low-risk neuroblastoma tumor sample, on the right, the high-risk sample. Low-risk tumors are highly infiltrated with B lymphocytes and CD-positive T lymphocytes, as you can see from the purple and blue staining, which is clearly present in the left panel, but absent in the right panel. So there's a big contrast here. When we look at the right column for the high-risk neuroblastoma patients, these are instead surrounded by a myeloid-rich environment, typically characterized by the presence of monocytes, neutrophils, and dendritic cells. So there's a clearly difference between these two samples. So I also want to take some time to acknowledge the people who have been doing this work and who are responsible for this project. So this is Annelies van Hemelrijk. She developed the panel. This was a very labor-intensive step. She put a lot of time in it. And this was under the supervision of Kat During and Charlotte Scott from the UGent and the VIB. So the next step in this project will be, first of all, to optimize further some key markers, like improving signal to noise for some targets. Then when this has been done, more samples will be run. And at the moment, we are also looking into a more in-depth analysis. So what we have now seen is a qualitative impression of the data, but we want to take this one step further and really go to a single cell analysis. So that's what we are currently working on. Next case I would like to share with you is focused on human moya-moya disease. This is a rare cerebrovascular disorder that is characterized histopathologically by the abnormal infiltration and proliferation of vascular smooth muscle cells in the intima layer of the arteries in the brain. The reason for this proliferation is unknown. Also unknown is the etiology of moya-moya. So the disease occurs worldwide, but prevalence is highest in East Asian countries. In this part of the world, the carrier mutation is found in 1 in 50 Japanese people, and however, only 1 in 10,000 will get the disease. So there are unknown extra environmental triggers needed to trigger the onset of the disease in people carrying the mutation. The team of Francis Impens, together with the University Hospital of Ghent, had assembled a cohort of patients that could be followed and from which samples can be obtained to do a thorough study on this yet very unexplored disease. Unfortunately, one of the patients passed away, but she donated her brain to science, and so on one of these samples it was possible to do a MACSima experiment. As I already mentioned, not a lot is known about this disease yet. So therefore, the choice was made to include an immuno-onco panel in the MACSima experiment. And besides that, based on single cell data, 6 extra targets were chosen to be added as custom antibodies to the panel. So eventually we stained for 146 protein targets using the Miltenyi panel and an additional 6 custom protein targets. What you can see here in the image is a subset of 6 markers, namely DAPI, RNF213, ACTA2, MMP2, FN1 and ISG15. This image nicely represents what an MMD artery looks like. We can see there is clear thickening of the lamina intima. This causes waving in the lamina elastica, and we see that the lumen is almost completely blocked by this thickening, so it's hard to imagine blood still flowing through this artery. So the idea and the purpose of the research is really to define which cell types can be found in these different layers, and for this MACSima and spatial proteomics seems like the good way to go. This slide shows the result of the cell segmentation, and already very high level of cell typing. So we see in purple the red blood cells, in red the immune cells that have infiltrated, and the green is showing all the other cell types. When comparing intima and media, we see that both layers stain positive for smooth muscle markers such as smooth muscle actin and myosin. However, it was also seen that the intima cells differ from the media cells in some markers. The group is still looking into detailed differences, but at large, the data confirms a synthetic phenotype of the smooth muscle cells in the intima versus a more contractile phenotype in the media. Such phenotypic switching is a known feature of vascular smooth muscle cells upon vessel injury, and it was previously already suggested to also occur in MMD. When looking at immune cells, we observed two large clusters. In blue, you see the cells that are present in the intima layer. These cells mainly stained positive for markers associated with myeloid cells. The cluster in green corresponds to immune cells in the adventitia that are mainly positive for lymphoid markers. Infiltration of T cells and macrophages in NMD arteries was previously reported, but our data suggests that T cells remain at the adventitia while macrophages are scattered more in the intima. So we are now still analyzing subtle differences in marker expression to more precisely define the phenotype of these immune cells. In conclusion, the MACSima experiment showed that there's a phenotypic switching of vascular smooth muscle cells that infiltrate the intima. An upregulation was observed of intimal FN1. This could potentially be used as a biomarker because it was also shown to leak into the plasma. Further, there was an infiltration of macrophages and antigen-presenting cells in the intima. If you want to know more details about this study or look at the results, this was a study performed by Professor Francis Impens, and there's a preprint available. This brings me to the last case study I want to share with you. It's one work performed by the team of Martin Gilliams, and they have been applying RNA-Sky to the liver. So they use the MACSima platform to combine the detection of RNA and protein on the same slice. So RNA-Sky is commercially released for FFPE tissues. It's using padlock probes in combination with rolling circle amplification to detect RNA specifically. So by now we all know how MACSima works for protein detection. So you have your section on a slide and then you get an iterative process of staining, imaging and erasing the signal. When we apply RNA-Sky, so when we want to use the platform to also detect RNA, we first do the RNA detection and then the protein detection. Martin and his team first used RNA-Sky in combination with protein detection on human FFPE liver samples. So they developed an RNA panel based on data they had acquired before via single cell or single nuclei RNA sequencing and available atlases. And they combined the detection of these 25 targets with the immuno-onco panel that is available from Miltenyi, and that is a fixed panel with 63 antibodies. Here you get an impression of the results. In purple we see protein staining, while the dots represent detection of RNA targets. We see here on the left the portal vein and on the right the central vein. And it's the what you see here, like the dots in dark blue, these show where KRT19 is present, and this is a marker that we only find around the portal vein, so not around the central vein. So this really shows the specificity of the signal. Also, we see that the red signal is predominantly present around the central vein, which also again confirms specificity of the signal. This is confirmed with what we see here in a zoomed in image from the previous slide. So the dark blue dots only present around the portal vein, and we can also appreciate that the purple staining marking endothelial cells is colocalizing with the light green dots, which are targeted against a marker of endothelial cells. Eventually, the lab also tried to do the combination of protein detection with RNA detection on fresh frozen samples from mouse. Again here, the panel of 25 RNA targets was used, but now in combination with a custom protein panel of 35 markers. The results are shown here, so we can again see protein staining indicated in purple and also in orange. We can clearly see again the dark blue dots being localized specifically around the portal vein, and this signal is not being found at the central vein as it should be. Zooming in, we can again confirm specificity. We see that for the cells stained in orange, which are the Kupfer cells, we see the light blue dots nicely present in these cells, which should be the case since the light blue marker is a marker for Kupfer cells. This can be confirmed with this other detailed image of the sample, and what we also can see again is that the purple staining of the endothelial cell colocalizes with the light green dots. In conclusion, our experiences with RNA-Sky have learned us that transcript detection is possible. You use an algorithm within the MACSiQ software to create the images we were just looking at. What is important to realize is that there is user input necessary to threshold for the spot detection, and in FFPE samples this worked very well. However, in fresh frozen samples we were confronted with a very low signal to noise ratio, which for us at the moment makes the technology mostly suited for FFPE tissues. For data analysis, this is still relatively complex and it requires steps to be performed outside of MACSiQ. This is also not easy to scale to multiple samples. So what is also very important to realize is that we noticed, and Miltenyi is also very much aware of that not all the antibodies work with the RNA-Sky protocol. So if you first do RNA detection and afterwards you apply a protein panel, you might see that some antibodies are not compatible with RNA detection, which do work when you just only do the protein detection. To overcome this, Miltenyi is providing information about antibodies in their catalog of which ones are compatible and which ones not.
So now it's time for the final remarks and conclusions for this webinar. So what is MACSima in a nutshell? What are the main pros and cons of the system and what are the main things to consider? So the platform has a lot of selling points, and one of them is definitely a very straightforward sample prep protocols. As a lab tech, I cannot appreciate them enough. They are really simple to follow. There are theoretically unlimited or very, very high number of markers that you can use to build your antibody panel. You're also flexible to add your own antibodies. There is a stable imaging, and the cost of the run and the consumables is not as high as for other platforms. What is also very interesting for us as a spatial catalyst is the compatibility with other spatial techniques, and we hope to explore this further in the nearest future. However, the platform also has its challenges, one of them being acquisition photo bleaching and throughput. MACSima may not be necessarily high throughput system, as it is not suitable for whole slide staining, so you will need to select some ROIs, otherwise the run will take too long. Also, at the moment, there is a manual image export and pre-processing. We are hoping that this will also change in the future. And the data analysis is also always a challenge, not only for MACSima or for all the spatial platforms, but again, we are learning, and this will also develop as we go along. And the most time-consuming steps in the MACSima workflow will be panel design, that we talked a lot during this webinar. There are a lot of things to consider and to think about, and it will take a while for the panel design. Data evaluation and data analysis also remain a very time-consuming steps. So if you are interested in the services that we provide, or you would like to learn more about how we run MACSima in service, you can send us an email. We would also like to use this opportunity to share some information about exciting conferences that we as Spatial Catalyst organize this summer. One of them is a Spatial Omics conference, which will happen this June in Bergisch in Belgium. And another one is Spatial Omics Summer School, which will also happen in Bergisch in Belgium. So thank you very much for your attention today.